We're delighted to have Louis Cohen with us today, who is a clinical investigator and associate attending at Rockefeller University, as well as being an assistant professor in gastroenterology and the Department of Medicine at the Icahn School of Medicine at Mount Sinai. Dr. Cohen got his Bachelor of Arts in Economics and Neuroscience from the University of Pennsylvania, and then was at Tufts for his MD, where he also completed an MA in Translational Research, also at the Tufts University School of Medicine. He did his residency and his chief residency in the Harvard system at Mass General, as well as Newton Wellesley hospitals, and then came to Mount Sinai and to Rockefeller to complete his GI fellowship, and also the Clinical Scholars Program at Rockefeller University. And for the trainees on the call, we just sent around information about that program to all of you, and I really hope that you'll take a look at it. For those of you interested in having a true physician scientist career, it's really a wonderful pathway to a productive career in a wonderful environment of investigation, that being Rockefeller University. And you'll see today that Dr. Cohen is such an exemplar of what can come from that kind of training and that kind of support. So think about that and review that information. Dr. Cohen is really a paradigm for me of two things. You know, we're all interested in advancing the field of medicine, advancing the care that we provide to patients. And how we get there really involves both answering fundamental questions about human disease, and you're going to hear about Dr. Cohen's contributions in that regard with respect to inflammatory bowel disease, and also advancing therapeutics. And here again, Dr. Cohen has played an important role in thinking about refractory IBD patients and how we provide care for them to get them better. He also, for me, has a special place in my heart because he is a scientific advisor to the Hermansky-Pudlak Syndrome Network and has been for a number of years, and has taken to heart a message you often hear me say, that rare diseases can help us better understand diseases that affect other populations. And so he's done great work in the HPS community, is a great friend to the network, and I'm deeply grateful to his contributions for these patients that I care about so much. Dr. Cohen, we're delighted to have you with us today, and welcome.
Thank you for the introduction, Sam. I appreciate that. You know, it's a pleasure to be here. I think this is a fascinating topic. You know, it's been a subject of my research, but you know, I also found that I found myself interested in human microbiome in part because as a clinician, as you said, you know, I kept seeing the literature as I was training exploding with study after study suggesting that the human microbiome had a role in just about everything. And so, you know, it led me to pause and think, you know, what is this data really telling me? You know, how do I understand this data? How do I sort of, as a physician, begin to think about its applications to my patients? And, you know, what's been extraordinary is watching the transition over the years as we've started to study the microbiome. And now I think we're on the cusp of its therapeutic potential. So, you know, I could also title this talk, Is the Microbiome for Real? And I think part of that is because, you know, you start to step back and you say, well, it's not possible, right? This thing that we've sort of started to study over the last few years has a role in every disease imaginable. You know, there has to be some balance here between what it's telling us and maybe some of the artifacts of being in a very early field. And so, you know, I have no disclosures at the moment. I'm hoping that'll change with time. But, you know, the goals today is I like to think about things again. You know, I think that my primary training is as a physician. And I think that as a physician, we see two types of studies, right? We see how we compare and contrast cohorts, and we also think about therapeutic studies. And so the goal today is so that we can all begin to understand how are these studies done and how are they done in the context of the human microbiome? Because I think, again, this is going to be an increasing part of our practice. And I think that as physicians, we have to understand these concepts if we're going to sort of embrace that and be, you know, the always intelligent consumers of data. So we'll start with kind of just an overview of what the human microbiome is. I call it kind of the fact versus fiction part. And then we'll transition into the idea of, well, how do we compare cohorts? And this is really the study of microbiome ecology. You know, what bacteria are there, And again, with a focus so that we can all look at this data that we've seen that's impressed us and be a little more.
Lost the sound.
Yeah, hold on, Norma. Louis, we've lost your audio. So if you just pause there and David will get us back together.
Dr. Cohen, you're unmuted. I think that must be something related to your devices, to your microphone.
Can people hear me?
Yeah.
Yeah.
Okay, good. So the microbiome, again, is, we think about it as being the bacteria plus an environment. So, as with many things in science, we stole a lot of what we're doing in humans from people that studied other microbiomes, such as the microbiome of the desert, microbiome of forests. And the other thing to remember is the microbiome isn't just bacteria. I'm going to say bacteria a lot today because that's what we've really studied the most of, but it's every organism, right? So humans are part of the microbiome of the land we're in, and hopefully we take care of it. Now, we've known forever, right, that the human microbiome has distinct areas, right? It has its own ecology in different places. And as physicians, this was always obvious to us, right? We saw bacteria in the blood. We could look at the bacteria that grew, and we knew that it came from the skin or it came from the bowel. But what started to happen is we started to change our thinking, right? I think that we always used to think that these bacteria were just there. And then suddenly we started to understand that they were doing something else, right? That they were providing cues to us. And then you started to sort of see people beginning to think about how are all these systems intertwined, what the bacteria are making, what we're making. And, I heard earlier about the idea of diet. How does this factor into all of these things together as a single system, as its own organ, if you will? And, one thing I think this led to was, the most fundamental misconception, which I'm going to correct right now. There is no such thing as a good bacteria. And we all know this. You would not want to take a single one of these bacteria and put them into your blood. The important thing to remember about our relationship with these bacteria is it's evolutionary. And the way that I always explain it, it's the same way of thinking about our skin and the sun, right? Our skin evolved to protect us from the sun. If we get too much sun, we get burned or we can develop skin cancer. But if we get just the right amount of sun, we can actually use that to our benefit and make things like vitamin D. We have the same relationship with these bacteria. They are all horrible. They're all terrible. They're all bad. We've evolved a way of keeping them separate from us. And if we do that in an efficient way, we can actually use the things that they make as cues to help our immune system function, our metabolic systems functions. So that's the sort of very first myth I'll correct. Never ever say that we have good bacteria or bad bacteria inside of us. They're all bad. And so as we began to understand this, the field sort of stepped back and said, well, wow, maybe these things are doing something. And how do we begin to think about it in a very orderly way? And I always love this. This is how it should have been done. First, we would start to study what bacteria are there. Then we would sort of say, well, what are they doing, right? What are some other functions that could be relevant to us? And once we understand that, we could understand how the human body responds to those functions. And then we'd be ready to move to the next step, right? What are the forces that maintain this relationship, right? So what are the forces that might shift the bacteria? How we respond to the bacteria? And then and only then, after painstaking research, could we move on to the final question, which is what makes us unique, right? Is there such a thing as a normal equilibrium? Is there such a thing as an equilibrium that gets put out of whack and that leads to disease? And then we did actually start with this, what are the identity of the microbes? And people had done this forever, right? We would go to places and we'd culture bacteria and we'd understand what's there. And so we began to think about doing this very systematically. As we sort of prepared, all of a sudden this happened, right? And this is why everyone's here today, that people kind of just jumped into comparing and contrasting cohorts, and they found all these crazy associations. And I specifically have 2012 on this slide for a reason I'll get to. And we saw this explosion in publications, right, leading up to 2012, where everything was about the microbiome and its role in different diseases. But we hadn't yet characterized what bacteria are there in a quote unquote normal state. We jumped straight from 1 to 5 because how could we not, right? It's so exciting. This is the thing that we really wanted to get to. And the reason we did comes from understanding how we study microbiome ecology and how that changed. Because I mentioned that forever we would just grow the bacteria. You'd go to a place, you'd culture it, you'd find the right growth medium, and then you'd get your bacteria and say that bacteria grows from there. But this always bothered us, right? Because how do we know what's missed? If you try to culture something and it doesn't grow, does that mean you just didn't know how to grow it? Or does it mean it's actually not there? And so this led to this idea of culture independent studies in the microbiome. So everything you're seeing in the literature when we compare cohorts are these culture independent studies. And this is basically the study of bacterial DNA. So when you look at all of this, the reason that things exploded prior to 2012 was that the government put forward a huge investment in the technologies to sequence bacterial DNA and to start to use that DNA as a surrogate to understand what are the bacteria that are there. And so what happened is right around that time, the cost of sequencing DNA became cheap. You know, in our lab, we have 2 DNA sequencers now. So this became a very trivial thing to do and led everybody to sort of get into the field and start to ask questions. And so for everyone to understand, you know, really when we talk about this idea of what bacteria are there, what defines different patient populations, this is the study of metagenomics. So a metagenome is the entirety of genetic material recovered from a sample. Right, a genome is from one organism. A metagenome is all the DNA we find, for instance, in stool. So then the next question becomes, how do we define a bacteria by a gene? Right, so we have all these different classifications of organisms. And basically this is this idea of molecular evolution, which I think a lot of people are familiar with, right? That genes undergo a rate of mutation, and that mutation leads to certain frequencies of different genes in the population. And that ultimately, the more similar 2 genes are across 2 different species, probably the more related those species are. Now, here's the important thing I want to make sure people understand again. When we think about all the bacteria that live inside of us, right? They're bacteria. They're probably pretty similar. So these are 2 major classes of bacteria, the most common classes that live inside of us, Bacteroidetes and Firmicutes. And so we might think that the relationship between them is when we think about, you know, our DNA and maybe that of monkeys. However, the reality is the relationship between these bacteria is like comparing us to corn, right? Never trivialize how long these bacteria have had a chance to get good at what they do. You know, we've had a couple 100,000 years, they've had 1 billion years to sort of try to figure out their strategies for coping with the environment. So the next question is, when we look at these studies, well, what gene are they assessing? And so this is something, again, people may start to learn more about. We look at this ribosomal gene, right? And we do that because basically every form of life has a ribosome. Every form of life has to make proteins. And so that provides a stepping off point by which we can start to compare and contrast different species, because we know that they're all going to have this similar gene. And the other thing that makes this gene important for us to study is it has 2 types of regions to it. And a lot of genes show this, right? They have regions that we call are conserved, meaning that if they mutate, the gene becomes non-functional, so they can't. And then there's other regions that are allowed that have that mutation, so it can get passed from generation to generation to generation. And this allows us to cheat. And the way that we cheat is that when we look at a sample that has a mixture, right, metagenomic, of all this different DNA, we can use one tag to identify that conserved region, right, because it's not allowed to mutate. So everybody's pretty similar. And then our machines will go forward and sequence into the variable region. And then we can take that variable region, and now we can start to identify, well, what are the different bacteria? And so we can compare it to gene sequences we have. And if it's really similar, we say that is that Clostridium, whatever. If it's a little less similar, we might say it's a related species. And if it's very little similarity, we're just saying all we might be able to say is it's a bacteria. And so again, this is the basis for almost everything you're seeing when they compare 2 different patient populations. Okay, so you've now learned how to do this research. So there are some problems though, okay? And this is what I think has led to a lot of the confusion about what the microbiome is and what it may be doing. So the first is that we have a lot of variable regions, right? And so what does that mean? Well, for instance, if you look here, this is one primer set where we looked at one variable region. And then this is another one where we looked at a different variable region. Every color is a different bacteria. And you can see just using 2 different sets of primers, suddenly we think that the bacteria in 2 different locations are vastly different. And they're the same bacteria, it's just how we're trying to study them. And again, if you look at the bottom of the page, that's 2012. So this was part of trying to build this foundation to create microbiome science. But we sort of jumped the gun and we got really excited before we did a lot of these fundamental studies. We now know that we can actually sequence the whole gene, and that helps us to get more accurate, or we can look at multiple variable regions together, and that lets us be more accurate. The other problem we had was our technology, right? So this is just looking at all the different ways that we can sequence DNA. So let's say we started with 100 base pairs of DNA. Every base pair may be predicted 100 times, right? Each of those predictions we call a read. But if you have a 1% error rate, which we had for a long time in the technology that we used to use, that means that every single base pair in your prediction is probably predicted wrong once. So what does that actually translate to? Well, thankfully now we don't have as much of an issue. Our ability to do this has gotten a lot better. But if you think about it like this, here's our original gene that we're trying to look at. These are all the reads, right? All the predictions. We have 2 errors here. In one case, we only had 1 prediction, right? And so that error gets transmitted. In the other case, we had multiple predictions, and we knew, in one case it was an A, in the other 2 cases it was T's, and so we know that it's likely a T, and we got that prediction right. But remember, gene similarity is species similarity. So this one error is a new species all of a sudden. And so that started to happen. We now know we can do more reads, we can sequence deeper, we call it, and we can get more precise that way. But this led to a huge problem, again, 2012. So this is basically a group saying, hey, we now know, if you look at the bottom of the page, there's multiple variable regions. We're going to do a good job and use different primers to study which ones are the most accurate. But then we see that for certain body sites, they correspond very well. But in other body sites, there's a huge difference, right? Some 10,000 unit difference. So what does that actually mean? Well, they're talking about this idea of an OTU, which is basically just a fancy term. It means operational taxonomy unit. You will see this in all the studies. And this is just a basic classification. We said, hey, if 2 gene sequences are 95% identical or more, they're the same species. They're an operational taxonomy unit. That we're just defining taxonomy by species, by sequence similarity. So now they're telling you that among patients, we see 30,000 different species. That's crazy. But it also was a big problem because it validated our belief system, right? When we had tried to grow bacteria for years, we thought people had maybe 100. Then with culture independent, using this new technology, we now see that people have thousands. But again, we had a problem. We now know using new sequencing methods, so this is shown here, again, in 2013 from one of my colleagues in Mount Sinai, for bacteria that aren't abundant, right? They're not a huge part of our population, you're not going to get a lot of sequencing reads. So you have a much higher likelihood of having an error and thinking we have something new when the reality is we don't. And now with new technologies for these rare bacteria, we can actually be a lot more accurate. And when we apply these new technologies, and this is my favorite part, we found out that looks like old school science was really still good. That maybe we probably have closer to 100 or 140 species or 160 species, somewhere around there. And we've now improved culture techniques where we can actually grow the vast majority of bacteria in our body. So when people say to you, oh, there's no way to grow them, how do we study them? That's not actually true. We can grow the majority of them. And it took us a while to understand that, but that's where the field is now going. So then finally 2012 hits and we got to the truth of the scenario. So this figure is about $200+ million. Every vertical line represents a patient, and each of these boxes is a different body site. And the colors represent bacteria in an incredibly, sky-high level grouping, right? So, we're not talking about a species, we're way up the chain in phyla. So when looking at bacteria from outer space, the take-home point from this slide is that no 2 people are the same. Everyone has different bacteria. And this was in healthy patients. So suddenly we're like, oh, this idea of ecology, how do you compare cohorts and start to identify differences if no one's the same, right? What is your actual baseline? What were these studies actually showing us before?
Losing weight.
What was that? So, now we start to think differently. Well, maybe it's not what bacteria are there. Maybe it's the ones that are kind of common to people, very prevalent, or maybe it's the ones that are very abundant, the ones that can become a huge part of our microbiome that are important. We're also starting to focus, maybe that species even wasn't good enough. We should really think about strain. And as physicians, we've known this, right? You can have E. coli or you can have E. coli 0157. 0157, your patients are miserable. Other E. colis, they don't care. So we know that strain level designations are critically important for function. And that's probably the thing that means the most to us. When we think about strain diversity, then our head really spins. So this is, again, from that same paper. And if you actually look at somebody versus someone else, so usually I tell everybody to look at the person next to them in the room. You know, this is a Zoom meeting, so you can just stare around the screen. No two people have the same strains. So you do not have, not only do you have a very different diversity of bacteria, but you don't even share one bacteria with the person next to you. Now, the other interesting thing is that your bacteria, the strains you have, are actually pretty stable over time. You know, that you see in this, that when you look at yourself versus yourself, a lot of the bacteria hang around. And that some bacteria may come and go, they kind of pass through, they may take hold for a little bit, but you have a core set of bacteria that are part of you. And that's what makes microbiome therapies very challenging, because these bacteria are good at living together. They do not want to let in foreigners. And we'll talk about that later. So what have we learned? Diversity is the norm. We have to be a little careful about older 16S analyses. We need to do a lot more sampling in order to understand what is that baseline variation in people. And we're starting to think differently about what make bacteria important. We also are starting to look at these other people that are coming along for the ride, right? The microbiome, the fungi, the viruses, the phages. And this data is coming out slowly. And again, these fields are now at the same infancy that the bacteria field was almost 10 years ago now. So when we look at this, I'm not trying to tell you that this is all made up, that these are all mistakes. It couldn't be further from the truth. This is a phenomenal start. These differences are real. What's been hard, though, is us understanding what the differences actually mean. And part of that has to do because the microbiome is so dynamic, right? It changes so fast. Now, maybe it's not new bacteria coming in, but the ratios of these bacteria will change incredibly quickly, which I'll show you. But I think at the end of the day, what we're starting to care more and more about is cause and effect. Do these changes mean something? Do these bacteria do something? Because ultimately we can't be that different, right? We're all people. You know, we all have hearts that function the same way, livers that function the same way. It can't be that our microbiomes are so vastly different in the way that they function. And so I think that this is kind of where the whole field is going now. So we started at 1, we jumped straight to 5. And so, you know, for the purpose of this talk, I want to kind of focus on how, when we think about looking at these studies, are we comparing and contrasting cohorts so we can, again, look at this research and read it with a critical eye and sort of say, well, what is the applicability to our patients? So just like any other sampling type study, right? We have to pick a cohort very carefully. We want to collect our samples, we have to process them, and then we analyze them. And just like we know in human trials, right, we want to control for confounding variables. That's the most important thing. You know, when you read a study, all you're thinking in your brain is, what are the confounders? The confounders are actually here. These steps that we kind of overlooked as we shot straight into human cohort studies. You know, what are the variables that we do that might just change the bacteria that have nothing to do with human physiology? And so when we think about our patient cohorts, right, we've learned there are powerful confounders. Diet is probably the biggest one. Age, the medications we take. And disease is an interesting confounder, because you could argue it's not just a confounder, but then it also suggests, and we'll get into this for therapies, the microbiome as a diagnostic, right? If disease predicts your microbiome, that's phenomenal, right? You can then maybe use the microbiome to predict disease. And so this is actually from a series of papers published in Nature, where they started to ask the question, well, you know, what do we see in the microbiome from some of the indigenous tribes, where their diet is incredibly routine versus the US. And we were able to predict, you know, people in the US are so different in terms of their microbiome compositions from people who haven't, again, seen antibiotics, who have a diet that's very, you know, homogeneous. The other question is, what about genetics, right? Do our genetics contribute to our microbiome? And so the take-home point of this slide is right here. If you look at dizygotic twins versus monozygotic twins, there's no real difference in their microbiome composition relative to each other. They're still very different from each other. So genetics, I think, doesn't have a role in shaping what bacteria are there, at least the way that we characterize bacteria now, but it does have a tremendous role in how we respond to these bacteria. And God knows my patients with inflammatory bowel disease are living proof that the way we respond to bacteria can have a huge impact on the way we feel. So now for sample collection, this is an important thing. I mentioned the microbiome is dynamic. So I'll walk you through this slide again from the Human Microbiome Project, our $200 million experiment. What we see here is this is the difference between the same sample just run on a machine twice. So, basically 0, if you just take one sample and you analyze it twice, yes, it's very similar to itself. If you look at the same person though, between 2 different visits, they start to become very different. And it's almost as different as you just look at 2 different people at the same time point. So your microbiome, even though the bacteria that are there are largely the same, their ratios are so different that it actually almost makes you look like a different person. So we really do need, if you want to understand what is someone's core microbiome, you probably have to do a lot of samples over time. Sample processing, thankfully, is something that we've worked through extensively. I can say confidently that we now, when we look at our data, when we process our data, that we've dealt with a lot of this sort of growing pains. DNA is very stable, but you have to be incredibly careful of other omics, right? So you see proteomic studies and metabolomic studies, take these with a grain of salt. This is a field that's in its infancy, even relative to where we are with the microbiome. And then when we think about analysis, this is, again, very sort of something I think that's familiar to us as physicians. So I'll use this paper as a demonstration. So, right, when we want to compare 2 groups, this is what you're taught, you convert and compare, right? You want to generate one statistic that allows you to encompass all the variables in those groups, and then you say, well, what is my p-value, right? Is that difference significant? So in the microbiome world, when I keep saying that people are very different, we're not talking about species composition, right? Because I told you the answer to that is 100% all the time. What we're actually thinking about is the diversity. How many different bacteria are there? What are their ratios? Are some very abundant? Maybe you have 100 bacteria, but 2 of them account for 90% of all the organisms. This is where we start to get into these population statistics. So we use these global measures of diversity or taxonomy. And so when you see microbiome studies, that's what they're telling you about. And that allows us to convert all this complex data into one statistic to say, yes, people in the US are very different. Our microbiome has a very different type of diversity than the microbiomes we see in other countries. We can also visualize this data like this, right? We use what's called a principal component analysis. And this basically looks at every variable and then just tries to predict, well, what are the variables that are key? What are the principal variables that predict the variation, right? It's a visualization tool. But if you look at it, it tells you that for one set of variables, we predict 25%. For another one, it's 6%. So that's 30-something%. There's still so much we don't know in terms of what is contributing to these differences. So what does it mean? That's the question, right? That's where we're at right now. These differences are real. We see them. But what does it mean? That's where we're trying to go. We know that populations are different. We know that these studies are informing us about something for human biology, but what we still don't know yet is this idea of cause versus effect. What do these changes translate to in humans? What we do know is that in mice, as we shift ecology, we can actually cause disease. We can diagnose disease. So the mouse models are really getting us excited that these things we're seeing in humans have meaning, but how do we translate that? And so, to kind of summarize this sort of last part, again, here are our confounders. We still have so much to do to understand how we respond to bacteria. We just don't know. But we do know a lot more now about the forces that are changing our equilibrium, the things that we have to control for in our studies. We still have no idea what normal means, right? We know we need a lot more patients and samples and time points, and we have to think differently about our outcomes, right? Ecology may be the wrong outcome. We really need to think about function. So the field has kind of put a stop on things right now. If you want to try to just find 2 patient cohorts and sequence them and submit it to a journal and think you'll get published, you probably won't, right? People have done a lot of this. The field now wants to return to basics, to get back to that linear sort of set of thoughts that I put out before, and really think about the second question, right? What do bacterial do? What are their functions? Because ultimately we think that's probably more important than just pure identity. That will tell you which bacteria and how they affect people, and what bacteria do we want to shape, maybe through our diet, and that those bacteria are important to those individuals. And so with this, we'll move to sort of the second half of the talk. which is to really think about therapeutics. So, I didn't necessarily in the first part, show you every single association and every cool thing. And part of that isn't because I don't think it's important, but because I really want you to have a framework where you can go and look at that literature to understand that too. And I'll talk about some of the disease associations that I think are the most meaningful. But again, I think that this is a field where as you start to see it, it should sort of give you a hint. And what I would argue, and I'll show you in a little bit, that judgment, those hints have been critical to how we've moved forward into this field of therapeutics. So just like everything, the clinicians are the ones that are making the discoveries and the thought process that's pushing the entire field forward. So we'll talk a little bit about how do we understand microbiome function. This is a lot about what my lab does. We try to identify discrete functions that we can then develop therapeutically. And why is this sort of your traditional pathway to drug discovery, but ultimately why the microbiome field has not followed this pathway. So where we're at right now is this is the microbiome field in its entirety, right? We have these really cool correlative studies in humans. Patient group A and patient group B have some difference. And that those differences, when we think about them in mouse models where we can really manipulate them, we have causative studies that by changing the bacteria, we affect an outcome. We cause a change in the mouse phenotype. I think the most interesting correlative studies we have in humans, we see changes in patients with cirrhosis. We see changes in patients who have steatohepatitis. In diabetes, we've developed models where we can actually look at those changes and maybe it's predictive of the medications they're taking, of them having diabetes. We also can have some hypotheses about how those changes could even be causative. We see it for obesity, for autism, for inflammatory bowel disease. And then for cancer, we've seen it in a couple interesting ways. One is that we have individual microbes that we've been able to detect using these techniques, and we actually can identify how they disrupt a cell's growth. And one of the more interesting ones, especially in the age of immunotherapy, we're seeing the fact that the bacteria that live inside of us can metabolize these drugs. For instance, some of the traditional chemotherapies we use, or they prime the immune system in a way where suddenly things like immunotherapy become much more effective or much less effective. And the reason I put these ones up here was that for all of these observations we've had in humans, they've actually translated well into mice. So these are ones where there really is sort of something here we're trying to understand, even if you're sort of, when people say to me, oh, you really think there's a connection between bacteria that live inside of you and things like autism or mood? Now my answer is, well, I don't necessarily think that they're causative in a disease like that. But when we look at the metabolites that bacteria make, they sure as heck make neurotransmitters. And so, there may be a role in how these certain diseases are manifested, right? So for a disease like inflammatory bowel disease, bacteria may be everything, right? That they are the trigger, they're the reason the disease starts, end of story. Whereas for other diseases, they may not start the disease, but they may very much have a role in how the disease progresses or its response to therapy. Unfortunately, even though we see these changes in people and we can recapitulate those changes in mice, it's not necessarily a linear connection, meaning that the bacteria we identify in humans that are doing something, when we put them into the mice, that's our answer. In fact, more often than not, that isn't the answer. So the real question is, when we think about moving to therapeutics, how do we even get there? If there's such a disconnect between what we're seeing in humans, what we're doing in mice, how do we even sort of begin these hypotheses to think about it? Well, a lot of it, again, has come from clinical observations. A lot of it has come from understanding the genetics of a disease. And then a lot of it has come from these early studies where we said, well, let's just start sequencing, right? Let's just start looking for differences. And hopefully there, we're going to find some kernel of truth. So now the next question is, we have this wonderful foundation, right? How do we move forward? And I think by moving forward, we have to answer the fundamental question, which is function, right? That if we think about it, somewhere between these correlative studies in humans and causative studies in mice, there is a central thread. And that thread is how those bacteria are either affecting the host cell or responding to a host cell. And in these interactions is the key to our therapies. These interactions are complex, as we discussed, right? They're not just a simple one-to-one, but they're happening over tons of different bacterial products, tons of things the host makes, not to mention everything that we're putting down the pipe. And buried in here, though, if you think about these words effectors and reporters, you could substitute it with treatment and diagnostic, right? If a bacteria affects a host cell, that's a treatment. If a bacteria can respond to changes in a host cell, that's a diagnostic. So a lot of people have poo-pooed things, right? There was this wonderful study in diabetes that showed patients with diabetes are so different than patients who don't. And then later on, the author said, well, what we actually discovered is the patients with diabetes, it's really just the fact they take metformin, right? It wasn't the diabetes, it was the drug they took that was impacting the microbiome. That's amazing. If you had a diagnostic in the clinic that told you your patients were actually taking the drug that you wanted, and it wasn't just relying on the A1C, that would be phenomenal. So bacteria, again, their job is to respond to everything that we're putting in our bodies. So hidden in there will be signals that I think will give us a profound insight into what our patients are doing, what they're eating, the medications they're taking, and also potentially the diseases that they may be at risk for or already have that our other diagnostic tests are not picking up. But for the remainder today, we'll talk about effectors, right? How bacteria affect a host cell, because this is my interest, and this is what I think is going to translate the best into treatments. So coming back to our $200 million figure, this was only part of it, right? There was a figure that they placed right underneath it. And so, the first figure told you that all the bacteria are different. And then underneath it, they put this figure, right? So the first one was the ecology. And then next, what they did was they said, well, we sequenced all these other genes, and we're going to map these genes back and predict to you what they do. And so these are all their metabolic pathways. And the authors didn't intend people to believe this, but I think it's what sort of people, when I've seen them discuss this article, they talk about, well, function's the same. But if you actually look at the functions that they were looking at, we talked about how for the bacterial phyla, they looked from outer space. This isn't just looking at outer space. This is just saying the bacteria are all bacteria, right? They make DNA, they process carbohydrates, they have to make, nucleotides for that DNA. So, there was no resolution here. It wasn't even meaningful the same way that looking at a bacterial phyla was meaningful. It was essentially just saying that bacteria are alive and they're bacteria. However, I think what it also tells us is that bacterial function in humans isn't that complex. It is pretty simple, but it's also almost entirely unknown, right? The important thing is that when people study bacteria in other environments, they're really complicated. Bacteria that live in the soil, they don't exactly have access to a lot of nutrients, right? So they've built up a huge genetic machinery to go out and take what's theirs. Bacteria and humans got a cushy job, right? They just sit there, we feed them, they feed off our mucus, like their life is easy. So their genomes are actually much smaller. And they don't have a huge portion of their genome dedicated to these sort of traditional things that we think about bacteria making that allow them to get what they deserve. And the reason that the Human Microbiome Project didn't intend to tell people that all the bacteria are the same is that they had this figure there too. So what they looked at was a bacteria in the mouth called Streptomyces. Everyone has this, right? This is one of these prevalent bacteria. So this has grown to be interesting. How does everyone have this bacteria when so many other bacteria people may not have? But in different people, right, we can see if we look at the abundance, some people have a lot of it, some people have a little of it. And then on the bottom, what they did was they said, well, is all the Streptomyces the same? And the answer was when they looked at functional markers, such as this ATPase unit or a specific choline binding protein, some Streptomyces had both, some of them had neither, some of them had one, some of them had the other. So even though, yes, everyone had this species, the function of this species across everybody was tremendously different. So, they never intended to tell everybody that all these bacteria are doing the same thing. It couldn't be further from the truth. But I think that the take-home point is that it's simple. And so the biggest problem we have right now is with our ability to predict function, right? So when you see these studies, oh, these bacteria are metabolizing this or doing this, you should be very cautious of this. So, right, when we did our sequencing and we wanted to think about what bacteria were there, the benefit we have is we have tremendous databases that allow us to say that this gene comes from this bacteria because we grew bacteria and we isolated their genome. For function, though, we don't have databases, right? To do function, you have to isolate a product, you have to study its effect on different cells. This could take one lab years, whereas to isolate a genome, you can do in minutes now. So our database size for function is very small. It's very poorly annotated. So as a result, we just sort of rely on predictions. And these predictions still just get you very much a general function, but not at the granular things that I think we're all interested in. How does bacteria A affect immune cell B? We don't know that yet. And so when we think about this, part of what makes it challenging is there's so many different ways to study bacterial function, right? Bacteria have things on their surface that bind to human cells. They secrete things that will bind to a receptor or affect the cell generally. They can process the metabolites we make and turn them into secondary metabolites. People have maybe heard about the idea of bile acids, and bacteria can metabolize them into secondary bile acids, and these can have a huge effect on the way that our gut functions. Unfortunately, we know very little about this. And so a lot of what the work that I've done in my lab is trying to dissect out these fine mechanisms, because ultimately I believe that that's where function begins and where therapies start. Unfortunately, this idea of approaching it from a very basic way, a bacteria makes a metabolite that hits a receptor. We then can take that bacteria that makes the metabolite and put it into mice, and then we can study how it goes into humans. This would be your real traditional drug discovery, right? We identify a metabolite, we test it for its function, we then test it in mouse models, then we move to humans. Unfortunately, this just isn't something that we've done in the microbiome. It's an incredibly uncommon way that we've discovered new therapies. What I think everyone hoped in the beginning was that we would discover therapies this way. We would compare and contrast the bacteria in two different patient populations. We would figure out that population A has either too much of bacteria A or too little of bacteria B. Then we could look at those bacteria in mice and realize that if you gave more of bacteria A, maybe you help to get rid of the disease or you cause the disease. And ultimately, we would then translate those observations into humans. Unfortunately, this system has been hampered by everything I showed you in the first half of the talk. That as we've compared and contrasted people to identify these key linchpin bacteria that are shifting us into a disease state, we almost never find them, right? That by thinking about a bacteria by the way that we're currently identifying them, it's been very hard for us to identify key bacteria and disease cohorts and make a difference. But what we have been able to do that's been interesting is, even though we can't figure out what these bacteria are doing, we can just take bacteria, we've isolated like a group of bacteria that we've isolated from humans, put them into mice, and suddenly we do see sometimes that those mice actually show us that phenotype, right? You take bacteria from humans that have colitis and you put them into a mice and the mouse gets colitis. Which bacteria is doing it? We don't know. Do all of them do it? No, but it's some unique combination of them, some special sauce. So that has actually been something that we've done a lot of. So the reality is a lot of the things that I'm going to talk about in this part of the talk have come from almost fortuitous discoveries through some bizarre hocus pocus where we've put bacteria into mice, they've changed how they've functioned, and then we've gone into humans. So for the final part, I think what we'll talk about is, well, as we've done this in mice, right? As we've sort of learned this, how are we translating this into therapies? Because these are things you're going to see in the clinic. So, I hope I haven't been too discouraging in that, we've kind of just stumbled through this field and found these things. It isn't quite that. We've had a lot of interesting thoughts. We've had a lot of interesting hypotheses, but it has been very fortuitous when sometimes those hypotheses have translated all the way through. So, I think when people are thinking about this idea of live biotherapeutics and, they hear this, for some of you, this may be a totally new term. For others of you, you may just sort of be translating it. Well, it's a live bio, and it's a therapy. You'd be like, well, don't we already have these? Don't I prescribe some for my patients, or I tell patients to go take them? And this is this myth of probiotics, okay? So probiotics are not live biotherapeutics, okay? The FDA created a new designation a little over a decade ago for the idea that we were going to use bacteria or another live organism in the diagnosis, cure, mitigation, or treatment of a disease. This is the key definition, right? Probiotics are not, at least the way that we think about them, the ones that you buy at GNC or Duane Reade, they're not intended for this use. The current definition for a probiotic, and so I think that when we say probiotics now, we have to be very careful. We're not talking about biotherapies, right? I think that we're talking about the ones that the FDA doesn't regulate. And they don't regulate them essentially because a patient can take them. And more importantly, the people marketing the probiotics aren't making a misrepresentation. So if you want to put a probiotic on the market, all you have to do is send the box to the FDA so that they can make sure that they agree that it's least safe and that you're not misrepresenting anything. Well, what can you represent in a probiotic? And this is the key part that I make sure my patients understand. you can claim in a probiotic what's called a structure function claim, that a probiotic maintains normal functioning of the body. So here's Floristore. I think this is one people may be very familiar with, right? What do they say? Our strain is larger, it's stronger. What does that even mean? It means nothing. It boosts our natural immune response. It strengthens the digestive balance. It supports your active lifestyle. Now, if you're a patient, you're like, man, I'd love a strong immune response. This is awesome. My balance is all out of whack. I got really bloated from those tacos last night. This to them sounds like a drug, but if you read very carefully, you realize they're not making any disease claims, right? They're just saying we're restoring a balance, because ultimately that means nothing. So, but it's not necessarily all myth. So this is what I would say is probably the most pivotal study in our understanding of the probiotics that are on the market, right? So these are the ones you can buy on the shelf. And so when you look through those names on the top left corner, you may recognize a lot of them, right? Lactobacillus this, or Bifidobacterium that. So this was published in Cell, and basically what they did, so this is the top right corner of the slide, they did two different things, right? They would give humans this combination of 11 probiotics that they put together, and then they went through the entire intestinal tract and they sampled, just to ask the question, do these probiotics actually make a difference? Do they colonize? And when they colonize, or if they colonize, or if they don't colonize, do they affect the genes, or sequencing, or response to bacteria? So they weren't trying to do anything about disease, but can we actually impact human physiology? And then they did the same exact thing in mice, right? They gavaged the bacteria, they looked for colonization, and they did it. So the first thing they learned is that one of these strains wasn't even what they said it was, right? But for the majority of them, they were. They did detect other contaminants. They weren't at a high level, but it was there. Now in mice, what was interesting is there was no difference whatsoever after they gave these probiotics in the ecology of those mice. So they didn't actually live there, or they didn't impact the existing ecology in a meaningful way. And there was no difference when we gave probiotics to mice in the host transcriptional pathways, right? So their response to probiotics measured in the most expensive way we can do it, we didn't see much of a difference. What was interesting in humans, if you took all humans together, we saw the same result, really no difference. However, and this is what the figure down below to the right showed, there was actually a subpopulation. There were some people where these bacteria actually could take hold, right? They could colonize. They did impact the diversity, right? That's this top group in green. And for those people, we actually saw an impact in transcriptional pathways in different areas, especially in the colon, less so in the small intestine. So what does this tell us? I think it gives us hope, right? That, even though these bacteria weren't selected for any other reason other than they grow in yogurt and they can get through an FDA designation just as being safe, in some cases, certain people can respond to them. And this is actually the truth of the microbiome and microbiome therapies. For therapies like this, it's going to be host factors are going to be equally important in terms of saying, well, which bacteria are the right fit for you? Because everyone has different genetics. We respond to bacteria differently. The bacteria we have there may be more or less permissive to other bacteria kind of coming in. And so now we need to understand if we're going to develop therapies, or I would argue certain types of therapies. So if you want to replace a missing bacteria, if you want to get rid of a bacteria, you're really going to need to understand these host factors and other bacterial factors that would allow us to do this. So on some level, people looked at this study and were like, yeah, probiotics are nonsense. I would argue that what this study tells you is that any bacteria in some people can actually make a change, right? And this is the signal that we need to say that, yes, what we're doing therapeutically is important. Now, when we take just sort of Cochrane studies, right, they look at everything. This is sort of hard, right? Because oftentimes we're using different probiotics, different doses. There's very little standardization, but a lot of the things that we've been interested in, that we're going to improve morbidity and mortality in preterm infants, right? There's no evidence. Can it treat IBD? There's no evidence. Now for necrotizing enterocolitis, this is sort of more new data. Maybe it's very, very low quality evidence. It's kind of a bit shaky. There could be a little benefit, giving these babies something. And then when we think about C. diff associated diarrhea, again, a little bit of sort of a little hokey, maybe in patients who are at a very, very high risk of recurrence of C. diff, giving them a probiotic could be helpful. What I would argue is that what you're seeing here, going from no effect to could be some effect, is really thinking about the microbiome in terms of when is it important that the microbiome is affecting the host, that those interactions are the key ones, to where it's probably just that microbe interactions, right? That just by changing just by throwing something into the system that perturbs it a little bit, you're giving a chance to impact an outcome. So for C. diff, to kind of explain this a little bit more, we've now learned, for instance, fecal transplant in C. diff, the top study, they filtered out all the bacteria. There are no bacteria in those transplants. And guess what? Patients did better. Was it maybe the viruses that got through the hypothesis? Was it the phages? Or again, it's just that in certain diseases, it's more microbe interaction. Just by shocking that microbe system, that's really what you need. It's not a function of putting the right bacteria in to affect the host in a specific way. The other thing we're learning about fecal transplant is, do we actually transplant anything? Do those bacteria stay? In some patients, the answer is no, and they do fine. In some patients, yes. Up to 70% of the bacteria may stick around. What's really interesting though, is if you look at the bacteria that stay in a transplant, they're awfully similar to the bacteria the patient had before, right? That we form this stable ecosystem. It gets screwed up in C. diff, but then when you try to replenish it, if I don't put the right ecosystem in that you wanted to see before, maybe the transplants don't work as well. So again, probiotics were never designed for this purpose. No one ever thought intelligently, oh, I'm going to take this bacteria in yogurt and that's the key. There are clearly signals though that probably using any bacteria can have an effect. So now we want to tailor that effect. How do we design a true probiotic where we say, this is something we're going to choose and we're going to make this happen? So this is the regulatory framework that we're now all watching play out in the literature. So the Center for Biologics Evaluation and Research said, this is a whole new thing, right? This isn't Remicade, this isn't aspirin. This is a whole new way of thinking about drug development. So they defined a live biotherapeutic. This is everything that's used to treat a disease. This is the designation that we should start to use in our vernacular. It contains a live organism. It's applicable to the prevention, treatment, or cure of a disease, and it's not a vaccine. So fecal transplant is a live biotherapeutic. Other strategies, microbiome therapies, I would argue, aren't necessarily, giving someone a prebiotic to grow out better populations, destroying bacteria with antibiotics, aren't live biotherapeutics. They would fall under traditional drug designations. What are the types of live biotherapeutics we're seeing coming in the pipeline? One is you can have a single organism, right? They think bacteria A has an effect. The other one is this magic sauce. Sometimes it's a mixture of organisms that they don't understand how they do this, but they do. Other times those organisms were tailored to have a specific function. However, again, one of the biggest problems we have is that oftentimes what we're seeing in mice doesn't really translate so well to humans. So there's been a third strategy that's been developed that's much more similar to what we would think. And this is this idea of a genetically modified organism. So this is actually just thinking about bacteria as a drug delivery system, right? That you engineer a bacteria to make a ton of some metabolite. You've studied that metabolite just like you would any other drug, but you think that by a bacteria going into the gut and delivering this metabolite right at the mucosa, that's where the therapy is going to come from. So now how do we validate these things? Well, how do we validate traditional small molecule drugs, right? We first identify a target that's important to a disease. We screen a library of small molecules to see which one affects that target. We then go through these iterations of formulation, right? We try to make it safer. We try to improve the bioavailability. We then can test it in models to look for efficacy and toxicity. And then finally it goes to humans where again, we look at efficacy and toxicity. So how do we validate live biotherapeutics? Is it just as simple as switching out these bacteria? And the answer is no. This is why the FDA has developed a whole new designation. And part of that has to do with because live biotherapeutics challenge this paradigm. We don't really know how they work. We don't really know how they do what they do because this fine granular data is missing for the vast majority of live biotherapeutics. And that's because we actually coupled drug discovery to the end result, right? We discovered these things because they work in mice. So we actually started almost towards the end. Now, because we don't know the mechanism, it becomes very hard to find new ones. But we thankfully have a lot of them that have made it already the way through. And we think that there's an advantage here because they can make so many different things, right? It's not just like taking one drug where you make one substance, maybe these complex things are a complex mixture of everything. So now when we think about formulation, formulation is a bit different. right? We still want to improve bioavailability and decrease toxicity, but we make it by fermenting, right? You got to grow these bacteria. They're live organisms, and that comes with challenges, right? Some of them are anaerobic. How do you maintain the right mixture of them? And we don't think about dosing in terms of milligrams. We think about dosing in terms of colony forming units. You know, how many live bacteria are you taking? This process is hard, right? Bacteria can change. And so you have to have a lot of controls to ensure the bacteria that you think you're putting in are the ones that actually were working in your model systems. There can be a lot of opportunities for contamination, and our strategies for making those pills are a little different, right? Maybe we want to direct a release to certain points, so that's not too dissimilar to what we think, but it's this idea of maybe in different points, the bacteria will colonize. Maybe it'll be they'll invade, they'll infiltrate, or maybe they'll just pass through, or maybe it'll be conditional, that some other factor has to happen for those bacteria to work. And that conditional work is something that then leads to a change in what we put in those capsules, right? Maybe we add things to the capsules to turn stuff on in the bacteria. So the toxicity, this is something that's come under the public scrutiny recently. So unfortunately, we thought this was incredibly safe. You know, fecal transplants in immunocompromised patients, everyone did fine. But then recently, we saw that what happened is in immunocompromised patients, there was a transfer of antibiotic resistance elements that actually led to fatalities. So now the FDA is very, very careful. The big concern is we don't want to put bacteria in that are going to teach other bacteria bad things, such as antibiotic resistance. So now our model systems, that's the easy part, right? We discovered it this way. But remember, the mouse microbiome is so different. So the FDA doesn't put a ton of validation on this. In general, we think that these organisms are safe, right? They were discovered in humans, they live in humans naturally. So they don't worry as much about it once you prove that they're not going to cause antibiotic issues. And they wanted you to get as fast as you can in humans. So that's why drug companies love this, because you go really quickly into humans. And this is really our first opportunity to say, well, how do the bacteria live? What is the toxicity? Can we actually pass the bacteria person to person? So just to end, I want to kind of just show a couple of things that we're going to see coming to market. So what are the live biotherapeutics that are in phase 3? So some of you guys may have heard about Ceres, right? This is, they're making it's a consortium of a dozen or so different microbial species. They won't tell anybody what. And it's to treat C. diff. They had a spectacular failure of their phase 2 study that almost shut down the whole field of live biotherapeutics. Basically, they got the dose wrong, and they also didn't define C. diff correctly. So they corrected that, and they just released in the last month their phase 3 data that showed a decrease in recurrence rate of 90% at 8 weeks compared to 59% in placebo. And just so you know, that's equivalent to doing a fecal transplant, but in this case, you just have to take some pills. Finch Therapeutics has the same thing for C. diff. Theirs is, they're calling it a full diversity microbiome drug. They don't tell you what's in their special sauce, just a lot of different bacteria. And they had a positive phase 2, but it was really like a phase 3 because it was a randomized placebo-controlled trial, just not at a huge number. And they saw prevention and recurrence at 75% versus 61%. So these are the ones that we're going to see come to the market pretty soon, depending on what the FDA has to say. Pretty soon could be a couple of years. So is there anything else in phase 2? Well, now we get into the inflammatory bowel disease world. So Vedanta has a consortium that was discovered by intelligently mixing and matching bacteria to induce T regulatory cells. And they're testing it now in phase 2 data for the treatment of Crohn's and ulcerative colitis. Ceres also has one that is a consortium. They didn't quite design it as intelligently as Vedanta, but also they're testing for ulcerative colitis. What do we see coming down the pipeline? We see lots of drugs coming through for neurologic disorders, dermatologic, cancers, metabolic disorders. I'm going to push through these slides for the sake of just showing you the final one, because we're running out of time. The last indication, we see things entering the phase 1, especially stuff to modulate checkpoint inhibitors. So this is for the treatment of melanoma. Food allergies, very exciting. And we have some drugs going in for obesity and metabolic syndrome. And so far, there's been no safety signal from any of these trials. So one of the big proponents for this type of therapy, that it's safe, appears to be playing true, although we're learning on the go, right? And that's what those studies from the New England Journal told us. So I'll stop here and thank you all for listening. And I hope this has sort of educated you about the field. How do we look at these studies? How do we think about them? And what are we going to start seeing coming into the clinic in the near future?
Thank you so much, Louis, for a wonderful talk. Burton, do you want to manage questions?
Absolutely. I will be, people are free to either unmute themselves to ask questions or write the questions in the chat, and I'll read them out for you.
Maybe I'll ask one to just as questions come in. Louis, I'm just curious, for the refractory IBD population, has anyone looked at that specific group of patients in terms of what it might teach us about their microbiome?
Yeah, so we're in the process of doing that right now. We have a program at Sinai for stem cell transplant for highly refractory Crohn's patients. We're looking at the microbiome.
Hi, this is during next. Hi.
Hi.
Sorry.
So the answer is we don't at the moment. I think what we're seeing in terms of some early signals is that their microbiome is actually very different than what we see even in, just sort of your active Crohn's patients. That from having had this disease for so long, they've really enriched for a bizarre set of bugs. And what's interesting is a lot of these bugs are actually really, really similar to what we see in the oral microbiome. So, the things that are connecting our body are starting to actually get out of whack. And so, I think that probably in patients with IBD, a critical component of future therapy, I don't know how much I believe that the microbiome is going to be able to take a true deep refractory patient and fix them. But I think the biggest thing is going to be, as our immunotherapies work, we have to find that right microbiome for those patients to give them a fighting chance that they're not just going to perpetuate the cycle. And this is unfortunately what we've seen in stem cell transplant. Right? There's no more powerful way of wiping out the immune system, but these patients don't stay healthy for 15 years, right? It's not like we turn back the clock to when they were a baby and they don't develop IBD until they're 20. They actually develop it again within a year or two. And so I think that a lot of that actually, it's going to be that inflection point. And so that's where our sampling has been the heaviest, where I think we may get a lot of interesting data. So I see a question, do hormones affect gut bugs? Can detailed diet histories help to determine why there's so much change in the same person? So those are great questions. Do hormones affect gut bugs? The answer is we definitely see changes in microbial populations and patients on different hormonal therapy. For me, I think the more interesting thing is that, do gut bugs make hormones? And the answer is probably yes. So, remember, our signaling systems, and this is a lot of the work that we've done, and I can show you just a quick slide to kind of highlight this from my lab. Our signaling systems in humans are almost identical to what we see in bacteria. So this is a slide. So this is one of the bacterial compounds we discovered. And we found out that this compound has a role maybe in inflammatory pathways. But if you actually look at signaling molecules that humans make, they're almost structurally identical. And we've actually expanded this, and we've seen this across a whole slew of things, right? That the reason these bacteria may be there is that they can share our language, right? They can respond to neurotransmitters, they can respond to hormones, and they can make things that look an awful lot like neurotransmitters and hormones. And so there's this, I don't know if it's a coevolution or what the mechanism is, or that this is this sort of shared signaling system that's been conserved, throughout all of evolution. So it's a fantastic question. It's something that I think that we've been phenomenally interested in. And it's allowed us to cheat a lot because once we find a bacterial metabolite, you can just go to all the human research and say, well, what is this metabolite going to do? And here it is. So, and I think I saw, I don't know if we have time for a final question. I think I saw one more.
I think, yeah, we do. There's one more question from Dr. Kojic.
Yeah, the interactions of toll-like receptors in the gut microbiome, the consequence on the immune system. Again, fascinating. So this is where, I mentioned before, right, that genetics don't have a big role in shaping microbial ecology. And again, the caveat being the way we understand it. Remember, I explained to you that if you just define a bacteria by its gene, everyone's different, right? So that isn't practical. But what we all know is that genetics of how we respond to bacteria, of which toll-like receptors are critical, are profoundly different, right? So we see changes in certain toll-like receptors that appear to accentuate or depress signaling events. And bacteria have a huge impact on this. So there was a phenomenal study that came from the Broad Institute years back that just showed you, we think about lipopolysaccharide, right? And I think that the common belief is lipopolysaccharide is one thing. Well, it's not. every bacteria has their own unique lipopolysaccharide. And certain of them, for bacteria that live inside of us, the really common ones like Bacteroides, their lipopolysaccharide interacts with toll-like receptors in a way that actually shuts down signaling, right? Because it's not just a simple receptor engagement, right? How that metabolite engages the receptor affects the receptor signaling. Whereas lipopolysaccharide from bacteria that are not that common to the gut don't have that effect. So that there's this phenomenon of our toll-like receptors have potentially evolved to decrease responses to bacteria that are common, right? And that makes sense, because if we tried to always fight these bacteria, right, toll-like receptors are part of our initial immune response. If we try to fight the bacteria that live inside of us all the time, it's like being pissed off at the ocean and kicking it and hoping that you get it to go away. It doesn't work. So, this is really this intensely important concept of what we call immune tolerance, right? And how does immune tolerance evolve? And what's fascinating is that not only does it evolve, but it appears to be specific for the types of bacteria we see inside of us. Again, bacteria were already there in the primordial soup. We evolved skin to protect us from the sun, and we evolved our immune system to help us deal with these bacteria there. So I think that that's why we see shared signaling systems. And I think that's also why we see these phenomena with things like toll-like receptors. So another great question.
If I may ask a question,
Sure.
Thank you for a wonderful talk, Dr. Cohen. So if going back to the original paradigm where, scientists were hoping to look at different persons with different diseases and find different bacteria and maybe try and modify that balance. And I think part of the reason that doesn't work is because, as you showed, even the same bacteria can have very different functions in different people. So has anybody tried and looked at transcriptomics? I know you mentioned that all of these other omics are in their infancy, but I feel like if we try and compare the function or the general transcriptome profile between these populations, we may have a better chance of knowing what is actually happening.
Yeah, I think it's a fantastic question. So, the two parts to that is a lot of people have tried to do bacterial transcriptomics. And this is where you have to sort of take things with a grain of salt. So what the studies will tell you is that the transcriptomics basically is just predictive of the genomics. And so they're like, well, why bother doing transcriptomics? We'll just do genomics. And that's because they're not asking the question that you're asking, right? How do those transcripts predict functions? So part of the problem is we still don't know how the genes, which encode for those transcripts, which encode for the proteins, function. So, when we look at these transcripts, all that they're able to do is map them back to the genes, and we don't know what the genes do. So therefore, we don't know what the proteins, the transcripts encode do. So that's the fundamental problem that interferes with what you're absolutely correct would be a phenomenal insight into bacterial function. The second problem with transcriptomics has to do with the time points, right? Transcripts are sort of very ephemeral. They'll kind of come, they get regulated differently. So sometimes, depending on when you decide to measure transcripts, you miss that event. So, when we try to understand how a bacterial metabolite affects one cell, if we want to measure transcripts, we might do one set at 2 hours, one set at 5 hours, one set at 10 hours, one set at 5 days. And so that's what makes a single time point for something that's really a dynamic process, like protein expression and gene transcripts, tricky, and it has made it challenging for the field because it requires a lot of resources. What we've done in my lab is actually sort of taken, kind of what you're saying, and we do metabolite screening. So what we do is we grow up lots of bacteria and we say, well, look, let's take the metabolites that we're able to produce in the lab. We isolate them. So they're not quite individual, but small groups of them. And then we screen them for functions that mean something. So for instance, there's a slide here looking at GPCRs, right? GPCRs are one of the most druggable targets. And so we've actually done screening where we've taken thousands of different bacterial metabolites and just said, can they interact with GPCRs? And so that's then allowed us to say, well, we don't care what bacteria are there. Let's look at the metabolites they're making, but we're going to assign function, right? We're going to start from a functional standpoint so that we know if patient A has metabolites A through Z, they would have the ability to affect these pathways in the intestine. And if patient B has metabolites X through Y, they would have the ability to affect these pathways in the intestine. And what we've seen, for instance, with one class of drugs, is this phenomenal relationship, right? So again, we see structural similarity. We see that even within a narrow family of lipids, each lipid change that bacteria have affects a different G protein coupled receptor. So if you have this one, you might be able to affect GPR119, which is involved in metabolism. This one affects S1PR4, which is an important dendritic cell regulator. This one affects the prostaglandin receptor. So it's important in wound healing. And again, the take home point from the earlier question is that if you look at the human ligand for these receptors, we see tremendous structural similarity. So again, it's this confluence of bacteria and humans talking. And from a functional standpoint, because transcriptomics has been hard, we've instead just said, well, what are the things we can do in the lab? And let's start to characterize those. And let's look at pathways that we think are therapeutically relevant, such as G protein coupled receptors.
And Dr. Cohen, thank you so much for a great talk and also for the important work you're continuing to do. And just really grateful to have you with us today. And all the best.
Thank you. It was a pleasure being here. All right, thanks again.